All set?
No.
No?
We have a half day. PDA day. Remember this? PDA. I know. No, it's PDA.
Remember, I was not correct. We don't need the broth culture, new broth culture for tomorrow. We're actually doing the motility next week. Okay. All we're doing tomorrow is we are looking at the results and recording results from all of the stuff you inoculated on Wednesday. So you should bring your camera or device that you take pictures with to get the pictures of the thioglycolate tubes, the plates, etc. And one word of caution, when you're taking those tubes, the thio tubes out of the incubators, try not to jostle them too much because you don't want to mess up the distribution of growth within those tubes. And make sure that you share the controls with your members of your group. What I'm going to have to do was the 25-degree gas pack was so full with knowns that we only had room for one set of, so full of unknowns that we only had room for one set of knowns. So I'll put those knowns from the gas pack over here. And so there won't be a complete set of group for the 37 ones. Okay. So make sure you look at those. Looks like things worked out pretty well. So, questions? Okay, we were talking about chemoheterotrophs. What characterizes a chemoheterotroph? What's your source of carbon? A chemical compound, and we've really been concentrating so far on organic chemical compounds, okay, as a source of energy. Where do they get their carbon? Organic compounds as well. So glucose could be a very commonly used substrate for chemoheterotrophic metabolism. We talked about aerobic respiration. We talked about anaerobic respiration. And then we were almost done talking about fermentation when the period ended. So this particular slide right up here is a fairly nice illustration of the diversity of types of fermentation that take place in different microbial species. Remember what you can ferment, and that includes what sugars you can use and what products you can use, is determined by what in the cell? What enzymes you have, which is determined by your genes. Okay? So it really is a reflection of your genetic makeup. And that's why these particular results of fermentation tests are useful in identifying different species. Anybody remember what test we used to determine the strong mixed acid fermentation, which really dramatically lowers the pH of the environment? Methyl red. That's the methyl red test. The one that determines this, the acetylmethylcarbinol being converted into 2,3-butadiol, is the Bogue-Sproskauer test. Okay. We're going to, in lab, after the lab exam, do some broth fermentation tests. And I talked a little bit about fermentation tubes at the end of the period. Basically, what the fermentation tubes consist of is a broth, and it's just pretty much tryptic soy broth. A phenol red indicator added to that broth. A little inverted tube stuck in the bottom of that broth tube. Remember what that one's called? That's called the Durham tube. And then in that mixture, we also put, in our case, one of 4 different fermentable sugars, potentially fermentable sugars. So we'll have a set of sucrose broth tubes, a set of glucose, a set of lactose, a set of maltose. Okay? Well, in order for fermentation to take place, sugars typically have to be converted into glucose, because glucose is then what goes through glycolysis, and glycolysis is the key pathway in fermentation, right? What's the relationship of sucrose to glucose? What kind of a sugar is sucrose? It is a disaccharide. It actually consists of a glucose and a fructose bonded together. So if you're going to be able to ferment the sugar, the first thing you have to be able to do is to hydrolyze that bond that joins the two sugars together in the disaccharide. You've got to have sucrase enzyme. Okay? If you don't have it, you can't ferment sugar. Okay? Glucose, if you've got the fermentative enzymes, you can ferment it obviously. It's ready to go through glycolysis. What kind of sugar is lactose? It is also a disaccharide. It is made up of glucose and galactose. You may know what maltose is? Another disaccharide. It's actually two glucoses bonded together. Maltose is called malt sugar. Okay? So for these disaccharides, if you don't have the appropriate enzyme that can hydrolyze those sugars into monosaccharides, you're not going to ferment. Okay? So that's diagnostic information. Assuming you can hydrolyze these then, what will you produce when you ferment these particular sugars? Well, it depends upon what species you are. Oftentimes, the fermentated products that are produced are acidic compounds. How could we determine if acidic fermentation has occurred using this system?
The broth will change color.
Phenol red will change color, and initially the medium is set up so it's a pretty neutral pH. And at that neutral and a basic pH, phenol red is a reddish color. We drop below 6.9, and now that phenol red turns color to a yellow color. So yellow tubes indicate some level of acidic fermentation. Do we know how acidic it is? No. Anything below 6.9 will tilt that color over to yellow. So that's all we know. Okay. Sometimes the fermentation will also produce gas. How are we going to determine the production of gas in those?
If the durum tube rises.
Yeah. What will happen is when you make these tubes and you autoclave them, it drives the air out of the durum tube, so the durum tube fills with liquid and sinks to the bottom. But then if those bacteria start producing little bubbles, I suppose theoretically you could see bubbles arising in the media, but they're so small you really can't. But if the bubbles start to congregate in the durum tube, what do they do to the liquid in there? Yeah, they displace the liquid, and then the tube will have an air pocket in it. And you're right, John, sometimes the air pocket's so extensive that that tube starts to float up. Okay? So we can tell a reasonable amount of information just from this simple fermentation broth test. We can tell whether the organism has the enzyme to hydrolyze the disaccharide to begin with. And then if it does, get some idea of the nature of the fermentated products that are produced. Okay? So that's something we will definitely do in lab. Fermentation has some interesting applications when we'll talk about medical microbiology. There is a very interesting organism, Clostridium perfringens, that causes gas gangrene. I mentioned gas gangrene when we did kind of our overview of some of the kinds of microorganisms when we did Bertie's manual. Clostridium is a bacillus bacteria that forms spores. There's one other really important thing about Clostridium. Anybody remember what it is? Clostridium is an obligate anaerobe. So these organisms, when they grow, ferment. One of the things that these ferment are proteins, and particularly muscle proteins. So if get a clostridial infection, and those bacteria start growing, they start chewing up your muscle proteins. This particular disease is also called Clostridial myonecrosis. It starts to necrotize your muscles, break them down. And in the fermentation process of fermenting muscle proteins, they produce CO2. What's that do to the tissue?
Raises acidity level.
Okay, we'll do that. Also,
Yeah, so
yeah, it starts swelling. It starts producing swelling, and that's one of the characteristics of gas gangrene is this necrotic, horribly swollen tissue. But then these organisms also, as a result of fermentation, produce some toxins. And those toxins contribute to further cell death, and that's one of the reasons why gangrene is a progressive disease. And what do you oftentimes have to do to halt the progression of gangrene? Amputation.
You said the CO2 makes the tissue swell?
Yep.
Okay.
Yep.
The toxins kill
And then the toxins kill cells. And when the cells are dead and you've got a whole bunch of necrotic tissue, that tends to become quickly anaerobic, so that the organisms can move into that newly created anaerobic environment and thrive there as well. So it's a pretty nasty disease. We'll talk more about that a little bit. Fermentation, particularly, remember we said that in fermentation, the final electron acceptor in fermentation is oftentimes an organic molecule. Sometimes it can be inorganic. No, it's always organic, sorry. Usually it produces acidic compounds or gas and things like that. But sometimes fermentation also produces things like vitamins and antibiotics. So there really are some interesting medical applications of the fermentation process. Here's a slide I'm not going to talk about, but I just want you to be aware of its existence. This is a nice slide to kind of summarize the differences between aerobic, anaerobic, and fermentation. The three pathways that are involved in chemoheterotrophic metabolism. Now, before we leave chemoheterotrophs entirely, I need to say a couple of words about nitrogen metabolism. Because up to this point, we've pretty much been assuming glucose is the starting compound for all of these metabolic pathways. Because glucose is what feeds into glycolysis, and all three pathways have glycolysis. But can you metabolize compounds other than sugars? Sure you can. And one of the things you can metabolize is nitrogen, particularly after you have hydrolyzed protein into amino acids. There are three reactions that you need to be aware of one of them will have some real consequences in just a minute when we talk about the nitrogen cycle, which is kind of an interesting way to illustrate all of the stuff we've been talking about previously. Okay, first reaction are amination reactions, or not amination, amination reactions. Amination reactions occur when some organic acid, like pyruvic acid, or it might be something like alpha-ketogluteric acid, which is one of the Krebs cycle compounds, or oxaloacetic acid, have an ammonium group added to them, and in the process we make an amino acid. So if you have a source of nitrogen, and you have the correct metabolic capabilities, the correct enzymes, you can convert sugars into amino acids. Bacteria do this all the time. Okay. Another thing that can happen is the process of transamination. In transamination, we take some kind of amino acid and react it with an organic acid, again, probably a Krebs cycle compound. And basically we can transfer the amino group from the amino acid to the organic acid, making a new amino acid. So in this particular case, aspartic acid is converted into glutamic acid. So once you can make a few basic amino acids, you can make the rest by this transamination process. But the one that's going to be most relevant to some of the discussions we'll have later on, especially in lab, is the process of deamination. Deamination involves taking an amino acid, like glutamic acid. Remember that amino acids are characterized by having a carboxyl group, an amino group, and some R group that varies from one amino acid to another. Remember? So if you deaminate an amino acid, you are removing that amino group and releasing ammonium into the environment. What does that do to your environment? Makes the pH more basic. Okay. When we had the fermentation broth tubes I was referring to, if your unknown doesn't have the enzyme to hydrolyze sucrose, therefore it can't ferment it, will your unknown grow in that fermentation broth tube?
Can you repeat the question?
Okay. If you've got an unknown and you stick it in that phenol red sucrose fermentation broth tube, and your unknown does not have sucrase, so it can't break the sucrose down, then it can't ferment it, right? But will it still grow in that broth tube? The broth is basically tryptic soy broth plus phenol red plus a durum tube and a sugar. Will it grow? Yeah. How's it growing? Because the tryptic soy broth has amino acids in it. And so your unknown would then deaminate the amino acids and form organic acids, which are part of the Krebs cycle. They're good to go. They'll grow. What will they do to the environment?
pH.
pH will rise. And how will you tell that? Actually, the phenol red gets a more purple color the higher the pH gets. So if some of the fermentation tubes, they won't turn yellow, they will actually turn a more purplish color. If they will ferment the sugar, if you leave them in the incubator long enough and they've used up all the sugar, what's going to happen? They'll turn yellow first, right? And then when they've used up the sugar, what will they do? They'll start working on the protein now, start producing ammonium, and eventually the pH will rise and the tube will eventually turn that purplish color. John, did you have a question?
Yeah, in the deamination, does it make another organic acid, like on the other side of it?
Well, the glutamic acid becomes alpha-ketoglutarate when it loses an amino group.
Okay, but is that an organic acid or is that an amino acid?
Well, you started with an amino acid, and it is turned into an organic acid plus an ammonia. Okay?
Do all organisms have all three capabilities? It just depends on which one they want to use first.
I would say all bacteria have all of these processes, and they're able to do this. Our cells, we have some transaminase abilities. We can transaminate some amino acids, not others. That's why there are certain amino acids that you must have in your diet because you can't form them from others. You know what those are called? Those are essential amino acids, yeah. Okay, so we have a limited transamination capability. Again, bacteria are more sophisticated than we are metabolically. Okay, so that's a lot of information about chemoheterotrophs. Let's just say a few words about these weird and wonderful chemoautotrophs.
Sorry, one more thing.
Go ahead.
So do they only use these when they don't have the enzymes to break down the sugars, right?
That's a good question. What determines whether you do deamination and then feed those organic acids into the Krebs cycle, or whether you do fermentation of the sugar first? Oftentimes there is a preference, and a lot of times it has to do with sugars, for example, repressing genes that code for some of these particular enzymes. Remember in the lac operon that if the glucose is present, then the organism will preferentially use the glucose and not the lactose? Yep, good question. Okay, chemoautotrophs. What is the source of energy for these organisms? Chemical compounds. And we're particularly interested in those that use organic chemical compounds. What is the source of carbon? Autotrophs. Inorganic carbon, CO2. So these are the organisms that can literally live entirely on inorganic substances. No requirement for sunlight. These are the rock eaters, if you like, the lithotrophs. A lot of these are the archaebacteria. Not all of them, but a lot of them are. And particularly the archaeans that are methanogens fall into this particular category. What the methanogens are capable of doing is combining a few hydrogens, not H plus, A2, sorry, with some CO2. And making methane, water, and ATP. These organisms have an electron transport system. And they can take hydrogens and remove an electron from the hydrogen atom. What happens when you take an electron away from a hydrogen? What's left? Proton. Okay? And then they pass those electrons down an electron transport chain. Here, the terminal electron acceptor is CO2. And if you add electrons to CO2, you make methane. And as a result of that, electron transport, you make ATP. The methane is oftentimes evolved from the cell in the form of swamp gas. If these are organisms living at the bottom of the swamp. Or cow gas, belching cows that have these methanogens in their rumen and produce methane as a result of the breakdown of sugars. There are an incredible population of methanogens in the ocean, and they have produced over time so much methane that has become buried under the ocean floor that we then, or oil companies then go and tap that methane for natural gas. Same thing happens in landfills. Okay, antigens are present in the soil and can convert those decaying molecules into methane, and we tap that in.
I'm sorry, under swamp gas, what is that word?
Oh, cow gas. Cow belches. Okay, sorry. Some of the methane that has become trapped under the ocean has become compressed into solid methane gas called methane hydrate. And you can go down under the ocean and excavate that stuff, and it looks like a big chunk of snow, but if you stick a match to it, it'll burn. One of the reasons that they're concerned about global climate change and oceans warming is that if the oceans start to warm too much, that methane hydrate starts to turn from a solid to a gas, and it would release tremendous amounts of methane gas into the atmosphere. And methane gas is a greenhouse gas, which traps more heat, increases the ocean warming, releasing more methane, traps more heat. What kind of a feedback system is that operating there? Positive feedback loop, okay? Keeps reinforcing and getting worse and worse. It makes itself worse. So there are some ecological consequences of these chemoautotrophic organisms, besides the fact that they are just kind of weird and wonderful organisms and violate that quote-unquote law that we've always told you, that microorganisms, or all life, depends directly or indirectly on the sun. These don't. Okay, let's do a little application study right now of what we've talked about so far, about chemoheterotrophs and chemoautotrophs. Let's talk about the nitrogen cycle. On our planet, what is the most common form of nitrogen in our worldwide ecosystem? N2. 78% of air is nitrogen gas. Is N2 very reactive? Very stable. Can it be used directly to make things like amino acids and nucleotides, which are nitrogen-requiring molecules? No. It's not a usable form of nitrogen to sustain plant and animal growth. What do we need? We need ammonium, really. And if you've got ammonium, then you can make amino acids and nucleotides. What's the process that gets us from N2 to ammonium? Oh. That's the nitrogen fixation. And the type of nitrogen fixation we're going to be most interested in is biological nitrogen fixation, which involves microorganisms. Remember, I did mention that nitrogen fixation can also occur by physical processes. Lightning, for example, can react with N2 gas and cause ammonium to be produced. A thunderstorm, literally in small quantities, fertilizes the soil, because the ammonium that's produced by the electrical discharge is washed down by the rain. We also talked about the fact that nitrogen fixation can occur both symbiotically and non-symbiotically. We mentioned the fact that cyanobacteria, those blue-green bacteria, are capable of non-symbiotic nitrogen fixation. They have this special enzyme that's needed for this reaction to occur called nitrogenase. Nitrogenase is very oxygen sensitive. It's inactive in the presence of oxygen. So there has to be some kind of a mechanism that sequesters or binds oxygen. And these particular bacteria will oftentimes form filaments, and there will be specialized little cells in that filament where oxygen is excluded. Remember the heterocyst? Botany? Symbiotic nitrogen fixation is an association between bacteria and the roots of legumes, peas, clover, alfalfa. And in the roots of these legumes are nodules. They look like tumors. And that's where the bacteria live. In this particular relationship, what do the bacteria get? Carbohydrate from photosynthesis from the plant. What do the plants get? Ammonium, produced by nitrogen fixation. But we've still got the problem that this nitrogenase is oxygen sensitive. So there is actually a pigment called leg hemoglobin that ties up the oxygen and prevents the nitrogenous enzyme from being destroyed. But unlike the hemoglobin in your body, once it binds it, it doesn't release it. So it grabs the oxygen and permanently inactivates it. Anytime you have a cycle, you always have to get back where you started from. So if we had only N2 being converted into ammonia, nitrogen fixation, what would happen to this eventually? Be depleted, right? So eventually we've got to get back to the start. That's why we call this a nitrogen cycle. Well, the nitrogen cycle is pretty complex. And it involves several different species of bacteria and several different metabolic pathways and strategies. Some of the organisms are facultative anaerobes. Some of them are chemoautotrophs. So what we want to do is kind of break down this nitrogen cycle a little bit and show you where various kinds of metabolism come into play. But if this cycle doesn't operate, we don't have life on this planet. Because if plants can't have access to ammonium, they won't grow. Nitrogen is a limiting factor to plant growth. And if plants don't grow, we don't survive. So the functioning of our worldwide ecosystem absolutely depends upon a properly functioning nitrogen cycle. One element of that, obviously, is nitrogen fixation. But there are some other elements to the nitrogen cycle as well that we've got to consider. Another is ammonification. Another is nitrification. And another is denitrification. Ammonification is pretty easily conceptually to understand. Ammonification is a process that adds ammonium to the soil, but not through nitrogen fixation. Ammonification adds ammonium to the soil through the process of decomposition. So what happens when plants die? They start getting broken down by microorganisms. And the protein eventually becomes recycled into organic acids and ammonia because the bacteria are using those organic acids as a source of energy. Probably aerobic respiration, maybe anaerobic respiration. So what reaction are we talking about here? They're deaminating those amino acids and releasing ammonia. Have you ever walked by roadkill before? Especially after it's been out for a while? What do you get a good strong whiff of if you smell that roadkill? Ammonia. Okay? Those microorganisms are breaking down the tissues of the plant or animal, converting the proteins to amino acids, the amino acids to organic acids, and the ammonium is being released back into the soil. Okay?
This may be off topic, but when like farmers, when they burn their fields, is that like anything to do with?
Well, I mean, I guess I'm not that familiar with the idea of burning the fields. My guess would be that would be to destroy weeds, weed seeds, things like that. I think that would be the reason you would do that. Now, the idea of crop rotation, which is an old, very well-practiced idea, involves alternating nitrogen-depleting crops like corn with nitrogen-fixing crops like clover, alfalfa, something like that, to replenish the nitrogen in the soil. I'm not absolutely sure about the burning, to be honest with you, but I don't think it has anything to do with this, because probably during the burning process, the ammonium would just be released as a gas.
When you do controlled burns, it's because you have nutrients trapped into plant matter, and it takes forever for the organic breakdown.
So you're just facilitating the decomposition.
Facilitate and burn it back down.
Okay. Okay. Nitrification is the first step in getting the ammonium back into N2 gas. This is the first step in completing the cycle. In nitrification, ammonium is oxidized into nitrate or nitrite. The organisms that do this are those chemoautotrophic organisms. So they're basically pulling electrons off of ammonium and producing nitrate or nitrite as byproducts. As it turns out, nitrite is kind of toxic. And so if a lot of nitrite accumulated in the soil, it would kill plants. Denitrification completes the process. It converts the nitrate back into atmospheric oxygen. Atmospheric N2, sorry. Denitrification involves the facultative anaerobes. So these are the ones that are using nitrate as terminal electron acceptors, and nitrate is eventually reduced into atmospheric nitrogen. Typically, the nitrate is not immediately in one step converted into N2 gas. It's usually a sequential process that involves several different microorganisms. So, for example, there are some bacteria that are facultative anaerobes that can use nitrate as a terminal electron acceptor in the electron transport system, and in the process, the nitrate becomes reduced into nitrite. Then a different bacteria uses nitrite as its terminal electron acceptor, and it's converted into nitric oxide. A different bacteria then converts this into this. And a different bacteria then finally converts the nitrous oxide into nitrogen gas. So the product of one bacteria is used by another bacteria. So there are several different species of bacteria. But we have to get from here back to here again to complete the nitrogen cycle. So these are critical organisms. Now, we're going to use this in lab. We're going to use this information in lab. And we're actually going to do a test to see if your organism has the ability to facultatively convert by anaerobic respiration nitrate into nitrite or nitrogen gas. And there's a test that we'll do later in the semester that will determine whether your organism has this particular ability. So again, we'll use a metabolic pathway as a way to differentiate between different species of microorganisms. Some can do it, some can't. If your organism is an obligate aerobe, is it going to be doing this stuff? No, because this is facultative. This is anaerobic respiration. This one's a little hard to see, but it's a great kind of summary and tells you some of the bacteria that are involved in the whole nitrogen cycle process. Okay? So we've got free nitrogen gas in the atmosphere. Nitrogen fixation can convert that into ammonia. At the same time, decomposition can turn dead and decaying plant animals by the process of deamination into ammonia. Okay? Here is the symbiotic nitrogen fixation that occurs in the nodules. These are the free-living, non-symbiotic nitrogen fixation. So two processes accumulate ammonia, okay, which can then be used in biosynthesis of new plant and animal tissue. But when they die, they return those nitrogen forms back to the soil in the form of ammonia. But we got to get from here back to here again to complete the cycle. So chemoautotrophs or lithotrophs, whichever you prefer, can then use this as a source of energy, and in the process make nitrites. Nitrites can then be converted into nitrates. This process is called nitrification. It adds nitrate to the soil. Nitrate can then be converted by anaerobic respiration back into N2 gas again. So one seemingly insignificant species of microorganism, if it's eliminated from this nitrogen cycle, can short circuit the nitrogen cycle. We're not going to talk about other cycles, but there are global sulfur cycles. There are global phosphorus cycles. There are global carbon cycles. And all of them involve this movement of the element through the environment and converting it from one form to another to sustain life. And the importance of microorganisms in these global recycling processes is absolutely essential. We cannot survive without those microorganisms who, through their metabolic activities, are able to do this indispensable function. So there are just a couple of other slides here that just kind of show you the nitrogen cycle. Oops. Another nutritional strategy is to be a photoautotroph. Source of energy? Sunlight. Source of carbon? Inorganic? CO2. Okay. All of the eukaryotic photosynthetic organisms, like plants, trees, shrubs, are all photoautotrophs. We also talked about cyanobacteria. They're microorganisms that are photoautotroph. They carry out oxygenic photosynthesis. And we also, when we did our Burgess overview, talked about green and purple sulfur bacteria. They are capable of oxygenic and oxygenic photosynthesis. We've already run through oxygenic photosynthesis, and you've had this before. Remember 2 photosystems, photosystem 2, photosystem 1. Okay. It produces ATP as the electrons move between photosystem 2 and photosystem 1. What's the other thing that has to be produced in the light-dependent reactions of photosynthesis besides ATP? Water is produced. Is it produced? Water's a reactant, right? What's the byproduct of water splitting? O2. Well, what's the other form of chemical energy besides ATP that's got to be produced? In order to convert CO2 into glucose, that's an uphill reaction. That's endergonic. We need ATP. What else do we need to convert this into this? We need hydrogen. Yeah. We need a source of reductant. That's NADPH. Remember, these 2 things, along with CO2, make glucose. What do we call that metabolic sequence? A complex pathway. It's a cycle. Krebs is respiration. Calvin cycle. Okay. The process of CO2 fixation or conversion into glucose is carried out by the Calvin cycle. So we'll spend just a couple of minutes on Monday talking about, again, reminding you about anoxygenic photosynthesis and how it differs. But then we'll briefly mention another weird organism. Weird. The photoheterotrophs. Don't judge? Okay. Wonderful organisms. Also Monday, we're going to start a new section. And that will deal with the control of microbial growth. So look on your syllabus. We're actually a little bit behind on the syllabus. But the next chapter is control of microbial growth. So that's what we'll be going Monday. So just come tomorrow with your photographic devices ready to